Today, 20th January 2022, first year microbiology, MLT first year microbiology. Topic we are already discussing ongoing process, sterilization and disinfection. It's continuation today. Last class yesterday we have to discussing in dry heat sterilization. Dry heat sterilization consists of red hot is used to sterilize metallic objects by holding them in flame till they are red hot. Red hot means is used to sterilizing metallic objects. Especially red hot is used to metallic objects by holding them in flame till they are red hot. మెటాలిక్తో ఉన్నటువంటి పరికరాలని మనం పట్టుకొని సూక్ష్మజీవులను స్టెరిలైజేషన్ చేయడానికి ఇక్కడ రెడ్ హాట్లో మనం ఉపయోగించడం అనేది జరుగుతుంది. ఎగ్జాంపుల్ inoculation loops, needles and tip of forceps wires, inoculation wires. Especially inoculation loops and wires and forceps used for the growth of microorganisms on media. Second thing, isolation of the microorganisms on the media. These are two processes you have to use for inoculation loops and needles as well as wires also. So why this process are used? The question is why this process are used? ఎందుకొరకు ఈ ప్రాసెస్ చేయాలి? This is a process of sterilization process. Second point is a dry heat sterilization. Dry heat is not moist. It's not consist of moist. It's only dry heat. Especially inoculation loops or inoculation needles or inoculation wires and forceps, you have to expose the flame, removing from the microorganisms. Clear? Removing from the microorganism. That means the sterilization of needle, the sterilization of inoculation loop, the sterilization of forceps. So you have to continue isolation of the organisms. Just you have to continue growth of the microorganisms. So this type of activity is used for red hot. Second thing, flaming. The article is passed over flame without allowing it to become red hot. The article is passed over flame without allowing to become red hot. Without red hot, you have to expose the articles, that is mouth of test tubes, glass slides, scalpels. In microbiological laboratory, we have to do the biochemical reactions in pathogenic microorganisms. Biochemical reactions, you have to inoculate microorganism. So that type of biochemical reactions, the expose of test tube the expose of mouth of test tube. Why? Because the mouth of test tube consists of a microorganisms. So unsterile test tube. So you have to sterilize. That means you have to expose the flame. That's why sterilization test tube, you You have to see in sterilization test tube. In glass slides, so microbiology laboratory, you have to do the staining purposes. You have to see in microorganisms on the microscope. uh, you have to see that process is staining process. Staining process consists of you have to take the glass slides. uh, Glass slides you have to stain. I mean, means a gram stain or Jaden stain or Arbor stain, and negative stain, capsular stain, or spore stain. You have to any one of the staining. You have to take first article is slide. So that slide consists of pathogenic microorganisms. So how to sterilize? Just you have to um, sterilize by flame. Sterilize by flame. In scalpels also. Scalpels you have to in biochemical microbiological laboratory you have to use it for scalpels. That scalpels also unsterilized. But how to sterilize? You have to by the flame of sterilization. Third one is incineration. First one is red hot. Second one is flaming. Third one is incineration. Incineration. Incineration means incineration, burning of the cloths or articles. Burning, especially this type of incineration, this type of method, mainly the process used for burning. Burning means కాల్చడం. కాల్చేసి పదార్థాలని మనం స్టెరిలైజేషన్ చేస్తాం. ఇన్ఫెక్టెడ్ డ్రెస్సింగ్స్, pathological material, animal carcasses, and plastic. So these type of all infected dressings, pathological materials, and animal carcasses, plastic, they are used for incineration. Then next heading is hot air oven. It's a very very important instrument for the microbiological laboratory. This is the most widely used method of sterilization by dry heat. This is an oven that has a double-walled chamber with a stout door. Stout door. This is an oven that has a double-walled chamber. Double-walled chamber with a stout door. The oven is usually heated by electricity and thermostatically controls. So this is an oven that has double-walled chamber. The hot air oven door is a double-walled. Then the oven is usually heated by electricity. The oven is usually heated by electricity. That's called thermostatically controlled. A fan is built inside the circuit air in the oven. A fan is built inside to circulate air in the oven. It should not be overloaded. It should not be overloaded. The material should be arranged in a manner that allows free circulation of air in between. The material should be arranged in a manner. Especially, why? Because this point is mentioned. The material should be arranged in a manner. Why? Because air circulation is easily. That can be removing from the pathogenic microorganisms. The temperature is, the holding period is 160 degrees centigrade for 1 hour. So how to operation that hot air oven? First point, glassware should be perfectly dry before being placed in the oven. Glassware should be perfectly dry before being placed in the oven. Then glassware should be wrapped in a craft paper. Glassware should be wrapped in a craft paper. Test tubes and flask should be plugged with cotton wool. Test tubes and flask should be plugged with cotton wool. Place the materials to be sterilized in the oven. Switch on the hot air oven. So you have to switch on hot air oven. So wait for 160 degrees centigrade for 1 hour. After 1 hour, switch off the hot air oven. The material has to be sterilized in very nicely. So in next class is a precautions of hot air oven. Thank you all of you.